All right, so I think we can probably go ahead and get started. June, would you mind just closing the door real quick? Thanks for taking the time to attend. Obviously, this session is Breaking Boundaries, How Next Generation Protein Sequencing is Shaping the Future of Proteomics. I'd like to start with just a quick round of thanks. First and foremost, thank you to Quantum-Si for sponsoring the panel session, allowing me to be the host. Thanks to the organizers of HUPO for putting on an amazing conference where you're bringing the best minds in proteomics together. And then also thank you to everyone in attendance, obviously, for taking the time. You don't have to be here right now, so I really appreciate, we really appreciate you guys taking the time. We at Quantum-Si, we really hope this conference is an opportunity to generate new ideas, to exchange existing ideas, and challenge yourself to think differently about your research. It's important, and it's our hope that this panel session will be part of that process. We want you to be able to ask new questions, have an opportunity to explore new tools that might enable you to ask new questions, and address any limitations that might exist with your current workflows. You guys are obviously very successful in proteomics, but we want to be a new tool to really enable you to take your proteomics work to new heights. So my name is Joel McDade. I have the pleasure of serving as the Director of Technical Sales at Quantum-Si, where I work with my colleagues at QSI and scientists like yourselves to identify areas where our technology could support your work and take your proteomics work to the next level. This session specifically will explore cutting-edge advancements in next-generation protein sequencing, tie that into some of the recent advancements that we've launched commercially, including pioneering protein barcoding kit to enable streamlined functional screening of proteoforms, as well as some advancements that we have around improving the input requirements for our kits and really improving the sequencing quality and the output. So following the presentation, we'll have a panel discussion. So I'll give a brief presentation, then following that, we'll have a panel discussion which will feature some of your colleagues, proteomics experts, Dr. Ben Garcia, Dr. Stephanie Cologna, and Dr. John Prensner. The panel will explore the implications of recent technology developments, invite them to talk about how next-generation protein sequencing and other technologies may fit into their work, and provide valuable insights into the changing landscape, specifically of single-molecule protein sequencing, and how that will change the future of proteomics. And so with that, I'll go ahead and get the presentation started. So again, Joel McDade, and we're going to be talking about protein sequencing today and how it's shaping the future of proteomics. So specifically, I want to talk through several key challenges in proteomics, what we feel at Quantum-Si are problems that need to be addressed. The first one is specifically how you efficiently engineer and screen proteins with novel function. The next will be how to characterize complicated, it's sort of a broad term, but complicated or closely related endogenous proteoforms. And then we'll introduce you guys to the Platinum Pro technology, of course, and we'll have a panel discussion and then Q&A. So this is not going to be a surprise to anybody at the HUPO conference, but proteins are complicated, right? Somehow in biology we go from 20,000 genes, easily read out on NGS these days, to 200,000 transcripts, to over 1 million proteoforms. And it's been really honestly humbling to listen to you guys talk as sort of a biologist. I'm really learning a lot about mass spectrometry and some of the other tools and some of the challenges that you face really around the breadth and depth of these proteoforms. It's an amazingly diverse set of biology that really we understand the tip of the iceberg from what I can gather. And this is really what motivates us at Quantum-Si, and we think that we can add significant value. Two of the key challenges that I'll talk about again are engineering and screening novel proteins. This process, again, you have this entire proteome to sift through, particularly in drug discovery, but also in academia, for these proteoforms that may have interesting properties, right? Whether that's binding, whether that's going to a tumor cell and killing it. But the process of screening through those proteoforms is actually extremely complex and cumbersome, right? So you have kind of the tried and true method of design, build, test cycle. I talk to a lot of synthetic biologists, and that's what they commonly say is design, build, test, right? That's a very slow process. And you have ways around that through automation, right? So large, expensive, unwieldy arrayed screens, and also, multiplex pooled screens. Now, multiplex pooled screens are an interesting one because it's very efficient, but there aren't really that many tools to do multiplex pooled screens with a protein level readout. So that's one of the things that we'll talk about today. But then further, when you're looking at endogenous proteoforms, characterizing endogenous proteoforms itself, or whether it's biomarkers or things driving disease, single amino acid variations, post-translational modifications, and low abundance proteoforms can be very challenging, right? Hopefully these aren't controversial statements, it's just kind of where we're at. And so, we think at Quantum SI that novel proteomics tools are going to be needed to address some of these challenges. We recognize the landscape of the proteomics field and recognize the important role of mass spectrometry, but we really do think that new technologies will be needed to address some of these problems. So I have the pleasure of introducing you to the first commercially available single molecule, single amino acid sequencer. Hopefully you guys saw it. If you haven't, I encourage you to check out booth 11 where we have one of the systems available for viewing. It is a benchtop single molecule, single amino acid sequencer, and it is designed for the molecular biologist in terms of ease of use. But for proteomics folks, it does provide very deep characterization of proteoforms and peptides. We are very excited about this technology. I've said a few of these things already, so I won't belabor it. But it really allows you to go deeply into proteoforms, again, at the single molecule, single amino acid level, in a way that has unparalleled ease of use, generally cost-effective, and very little expertise required. So if I have a show of hands, how many of you have lifted a pipette before? Come on, guys. Yeah, not in a while, right? Yeah, anybody that can lift a pipette can operate the system, right? And so it's really unparalleled in terms of its ease of use, and it provides extremely deep interrogation of proteoforms. So how this technology generally works, and I recognize this is like a very simplistic view, we have a set of consumable kits that we provide, library prep kits, we call it, when you're just sequencing proteins of interest. And then a barcoding kit when you actually generate a barcoded protein and you want to perform a screen and isolate a barcode and use that as the readout. I'll get more into that in a second, but those are all commercially available from us, as well as a sequencing kit, which allows you to obviously do the sequencing. So what you'll do is you'll take this prepare step, you'll isolate your peptides or barcodes, you'll generate a functionalized library. That library of peptides you then flow onto a chip. The chip is, we have it on display at booth 11, but it's a small microchip. A lot of IP went into it, but it's shown here pretty straightforward. So you'll flow the peptide library on. After functionalizing the peptides will bind, sit down in individual wells. I'll go through the mechanism in a second. But then you essentially set it and forget it, so to speak, right? You put it in the sequencer, you press go on the touch screen, and then on the back end is analyzed data. So all of the sequencing and the data analysis happens on the system or in the cloud. And so you don't have to do any post-run sort of analysis if you don't want to. You can export some of the data to do deeper characterization if you'd like, but it's pretty straightforward. So this was all published in a science paper in 2022. Some of you may have read it. If not, I'll give sort of a brief synopsis. So essentially what we're doing at Quantum SI, again, is you take your protein or proteins of interest, you digest them to make a library, you flow those onto a chip, and then each individual well will house a peptide. That peptide is then recognized by a series of what we call recognizers, which go through on and off binding on the order of seconds. You have a laser, a 1-watt laser that illuminates on the order of nanoseconds. And these binding events occur on the order of seconds, and what they end up generating is something that looks like this, where this is a single well on a single chip with a single peptide, and you essentially get a nice big signal here. Each of these individual peaks, if you stretch this out in time, you'll see the on binding, you'll see the off binding, right? And you'll see an intensity component, you'll see a decay component, and you'll see what we call a pulse duration component. And the pulse duration component is essentially how long the recognizer is being bound. So for each, just to let this point set in, for each individual amino acid, you're getting hundreds of binding events suggesting that amino acid is there, right? And so this is an extremely data-rich technology. If you think about what a read means on this, versus what a read means on something like next generation sequencing for DNA. But going on so on and so forth down the peptide chain, what happens is aminopeptidases come in, they're tuned to clip off amino acids at about an order of 10 or so minutes. And so you'll see the R binding and you'll see a bunch of pulsing. You cleave that amino acid off, going down the chain, you see L, notice the difference in intensity here, so on and so forth down to I and F. That data is actually captured by the technology and converted to what we call a kinetic signature plot. And that kinetic signature plot is essentially the software saying there's enough pulsing here that's distinct that we think that now this is actually going to be an R, right? So, and each of these have, again, these variables called pulse durations, which on average are unique to the amino acid in the sequence context. So you've set up your run in the software, you've run the system. We have now what's called a primary analysis, which is basically taking all of those recognition segments and calling them for the recognizer that bound. You can do a couple different things here. You can do what we call a peptide alignment, which is basically like, I know I produce recombinant IL-6. Please align all of the reads against IL-6, right? Or variants of IL-6, whatever it is. If you know what you're looking for, this is the route. If you don't know what you're looking for, you're like, look, man, I have no idea. I did a pull down with my antibody. I want to know what it binds. We have a technology called protein inference. This sort of conceptually will probably be very similar or very, very familiar to the mass spec folks in the room. But you essentially rank order the likelihood of a particular protein by the number of peptides you've identified, the number of reads you've identified, the uniqueness across the proteome, so on and so forth. So it gives you the answer, is essentially the workflow. So two different workflows, one for sort of confirmatory work, one for more discovery work. All right, so what does the data look like? So in terms of the data, here's an example of one particular protein, PDL1. PDL1, when you digest it and you sequence it on platinum, it generates a series of peptides here. I should mention that this is a lyc-based digestion. And so, the peptide profile will be determined by its digestion with lyc. And what you get is a lot of information. So you get the total number of reads that are attributed to a particular protein. You also get the number of peptides that you've identified. And these, I should say, these are alignments. So these are like, it's gotten sequenced, there are recognition segments, we know it's a high quality read, that read has been aligned to a reference sequence and the alignment matches. If any of these recognition segments don't match the proteoform, it won't get aligned, and we have a very strict bioinformatic algorithm for that. And it also comes with an FDR, so you have a sort of a confidence score, a number of alignments for a particular peptide, and then going down the amino acid, here's an example of a peptide, RFNEL, which is essentially showing you the coverage across the peptide, the coverage across each amino acid, but then some really in the weeds information like pulse duration, which again is indicative of that particular amino acid. See a nice histogram there. But also other things like interpulse duration, how long it takes for the enzymes to actually cleave off the amino acid. There's a ton of data here, and it's all sort of at your fingertips. This is generally what's shown, this is shown as well, and then you can export the data in Excel if you need to. We're applying this across a variety of workflows into a variety of different application areas. So things like confirming proteins, confirming proteoforms, protein variants. If you travel to our website, you'll see a lot of information around that. We want to make it functional, so things like co-IPs, we're working on a lot internally and applying that primarily to antibody biologics work. In-gel digest, and I have some of this data in this presentation, but in-gel digest we're applying a lot to things like biomarkers, biomarker studies, and then obviously protein barcoding, which I mentioned before. So the first sort of use case I want to go through is this concept of designing and engineering and then screening proteins. And so really, like I said before, you have a variety of proteoforms that you can use for any particular purpose, but you have to be able to screen those proteoforms. And this is traditionally very challenging, and so you can do it one by one. So I want to engineer these variants, I want to see how they behave in my screen. It's slow, it's cumbersome, it's variable. No one wants to do this, but we have to do it sometimes. Obviously, companies and others have really gotten savvy with generating automated solutions for some of this stuff, right? Automated, plasmid synthesis, automated plasmid transfection, automated Western blots, et cetera. But these are generally expensive, can be technically challenging, and they're variable. In the case of pooled multiplex screens, so you take X amount of variants, 100 variants, whatever it is, you pool them into a single pot, you do a screen, you do the bake-off. That is really, I think, where the money is made in terms of biologists' ability to operate at scale. The challenge with this is it's often indirect, and so you'll see a case study that I'll present where there's a group that wants to screen mRNA-based therapeutics, but the problem is the protein is exactly the same. So they're varying mRNA chemistries, they're varying ITRs, they're varying UTRs, they're varying caps. And so the readout doesn't allow you to multiplex unless you use NGS, but we want to do protein level work, right? We want to study proteins. And so that's a real problem for that field. And so we recognize this at Quantum SI, and so what we developed was called a barcoding kit. And that barcoding kit, essentially what it does, it allows users to generate highly sequenceable, right? So they're sequenceable and designed a priori to be compatible with the technology. You design these barcodes and you rationally design them to be short, the masses to be similar, the charges to be similar. So biochemically they behave very similarly, but you allow it to uniquely identify proteins and read it out on the sequencer. So now for things like antibody uptake or things like secretion, what you can do is you engineer these barcodes for each variant, you do a multiplex pooled screen, and your readout is direct and it's highly sequenceable on Platinum. That's the idea. And so we have a paper that we published recently in bioRxiv, and so this describes the technology. I won't do it ad nauseum right now. I'd encourage you guys to visit our poster, poster number 24. It's up today. I'll be standing there, hoping you guys stop by. But there's a barcode construct design which people can use and iterate off of, which allows you to quickly generate barcoded proteins. Obviously, you have whatever screen you would want to do, but then there's a protocol that's essentially a 3-hour protocol to isolate these barcoded proteins and then to cleave off the barcodes, and then, of course, sequence them on Platinum. It leverages, obviously, the Platinum sequencing technology, and that's really, really a straightforward workflow with a direct protein-level readout. So this is the case study. I've sort of introduced it already. It's a biotech based out of Boston. We partnered with them to really kick the tires on this for in vivo screening of mRNA. So essentially, which of these mRNA candidates makes the most protein is the one that I want to use to generate vaccines. We also have a lot of interest in this application from pharma, primarily because monkeys are extremely expensive, and wouldn't it be nice if we could cut the number of monkeys by an order of magnitude, let's say. So there's a lot of interest there, but it's a problem, right? Like they don't have a way to screen this today. And therefore what they do is they resort to, okay, is my mRNA delivered? And is it delivered is a question that's different than is it translated, right? And then also is it, it's not biologically relevant. So you could pick the wrong candidate, I won't get into the papers, but you could pick the wrong candidate by looking at this metric, and they want to solve that problem. And then, so obviously the goal I've stated several times, right? And so the goal is really to move to a direct protein level readout in vitro, in vivo, et cetera. We have a case study on this. This is the general workflow where they mix mRNAs and LNPs injected into mice. And then that becomes translated. So you essentially have the bake-off of all these candidates producing proteins. And again, the protein is generally identical with the exception of the barcode. And then you take that barcode, you enrich it, and then you sequence it out on Platinum, and that's the idea. And the poster has much more information about this and goes into the details of the mechanism and that sort of thing, and the sensitivity and so on and so forth. So again, I'd encourage you guys to read the poster. More traditionally, again, characterizing proteoforms, I think, is obviously a big challenge, and we think the Platinum adds value there. I won't go through this again, but suffice it to say, again, you're taking protein or proteins of interest, you're digesting them into peptides, and then you're getting protein level readout, peptide level readout, and amino acid level readout. So we've been thinking a lot about biomarker work. So obviously you've got technologies like Olink and things like that doing great technology doing high broad scope which of these particular proteins is up or down, right? We think this is a great technology for going deeper, so really going into the biomarker space and being able to look at, okay, this particular protein is up, but which isoform of that protein is up, right? What PTMs might be there. So really going deeply into that work. And so here's a workflow that we've been working on and developed internally where you can essentially take an SDS page gel, run serum on an SDS page gel, look at bands that might differ, sequence them out on platinum, here's just an example of traces, and then essentially query the system to say, okay, well, which one of these proteins is this, right? And which isoform, because you could, again, all you have to do is iterate the reference sequence, which isoform is it, right? So you can go pretty deeply. Just quickly, since some of you may have seen that data, we've done some work internally to really drive down the limit of detection within this system, because one of the challenges you guys know already with biomarkers is they can be really low abundance. And so what we've done is we've essentially started working on this optimized protocol where you can get down to about a nanogram of material out of an SDS page gel and be able to sequence it and be able to predict with certainty what protein it is. In this case, albumin, but of course, we plan on expanding that. That work is actually in an app note, which we recently launched, and so I'd encourage you guys to check that out, where we've basically demonstrated that capability within the tech note. But we also have a couple case studies that are relevant for this audience. The first of which is with Gloria Shankman at UVA, where they're doing a lot of RNA-seq, and they're identifying variants of a protein called tropomyosin that, when mutated, you can have a point mutation that results in disease, but a lot of times those point mutations are actually isobaric, right? So it makes them challenging to study with traditional technologies, and so their vision for this technology, they do have one in the lab, there's a picture, where they want to use orthogonal methods to study these, right? Not as a direct replacement necessarily, not as, but just really as an orthogonal method that may behave, quite well in some of these situations. So this is published in bioRxiv, and really what it's looking at is some of these isobaric changes that, going from one isoform to the other can actually result in disease, but we're able to resolve those very quickly on the technology because isoleucine looks very different than leucine, for example. And that's just sort of one of the core tenets of the technology. But also some of the alternative splicing can get pretty complex, and so being able to resolve some of these closely related isoforms that they see via RT-PCR, and they want to know, okay, well, it's expressed, but is it actually there at the protein level? And then finally, looking at starting to go into the world of post-translational modifications. Neil Kelleher honestly kind of blew my mind at the beginning of this whole symposium because he mentioned that tau has potentially 2 to the 65th different phosphorylation states. Right? That is a really, really, really complicated problem. So I think, to me, what that says is we need tools, different kinds of tools, to be able to study some of these things. And so here is just one example of looking at the distribution of phosphorylated tyrosine that's relevant for TPM2. But I encourage you guys to check out the publication. And finally, we have a publication in bioRxiv with Neil Kelleher looking at the sort of the complementarity of top-down mass spec with platinum from a bottom-up perspective. And so what we saw when we did this work, which is on recombinant IL-6, is essentially the overlap between the coverage between the two technologies was complementary. Right? And so we think that there's a space to complement mass spectrometry for this type of work moving forward. So to summarize, I want to get you guys to the panel. I want to have you guys listen to your peers. We're working on a lot on the application side. I'll get to the technology roadmap in a minute, but anybody that's been following us, we've had a dizzying amount of content come out on the platform that is commercially available today. So we're working really, really hard from a workflow perspective to make this technology useful for you guys and your colleagues. And then we're also, applying this to a variety of areas, right? Vaccines, antibody biologics, both of which we already have users actually in the field using this technology for, protein engineering, and then peptide therapeutics. Again, where we've been and where we're going, I've been with the company about 2 years, and truthfully, it's really incredible as a professional to be a part of something that's moving this fast. From the time the publication came out in 2022 to the first commercial sequencer being available to the first kit being available, you can see kind of the trajectory here. We're already at V3 of our sequencing kit. We're at V2 of our library prep kit, and now we just launched Platinum Pro, which is shown here. And then 2025 and beyond, there's an investor deck that you guys can see that kind of lays out in a bit more color where we're going. But yeah, I mean, as our early adopters are benefiting from this, right? And so, if you bought the technology back in 2022 or 2023, you've already gone through several iterations of the kit, and it's really improved the output. And I think, overall, we think that the market's really recognizing this, and we're really, moving forward and moving fast. And so with that, I will say thank you, and we can transition over to the panel portion. We'll hold all questions to the end. I will try to keep some time for Q&A. So from there, I think, what I want to do is move over to the panel session again. I'll just do quick introductions for each of those that are in the panel. Start with John Prensner from the University of Michigan, who's actually kind of funny. I was going through his bio, and I realized that we did our PhD and graduated from Michigan at the same time, and then moved out to Boston and started our postdoc at the same time. Mine ended sooner than his did, but yeah, so, I can go through the whole litany of sort of accomplishments and things like that. He's bachelor's degree in English from Tufts University, MD and PhD from University of Michigan, focused primarily on long non-coding RNAs in cancer. He's got a residency training at Children's Hospital in Boston, and then postdoctoral work with Dr. Todd Golub at the Broad Institute of MIT and Harvard. And then his lab focuses primarily on the molecular basis of cancer with a particular interest in pediatric brain cancers. And so I will also mention that he's in the process of onboarding Platinum, and we're really excited to have him. Thank you, John. Stephanie Cologna. So Stephanie joined UIC in 2015 and was named as the Associate Vice Chancellor for Research in 2021. She's a faculty member in the Department of Chemistry, and appointments in Biological Sciences, and the graduate program in Neuroscience. Her research interests lie at the intersection between analytical chemistry, biochemistry, and molecular biology. She has her bachelor's degree in chemistry from Arizona and a PhD in chemistry from Texas A&M. So. Thank you.
Thank you.
And then Dr. Ben Garcia. So I would imagine many of you know Dr. Ben Garcia as the president-elect of HUPO. He's also the Raymond Witkoff Distinguished Professor and head of the Biochemistry Department at WashU. And he's recognized with many awards, including the American Society for Mass Spectrometry, the Beeman Medal. He's at the forefront of academic and industry approaches to quantitative proteomics. So please join me in welcoming our panelists. Make sure this is on here. So what we'll do here is we just have some questions that we've prompted the panel with. We'll go through those. My hope is that it takes 20 or so minutes, and then we'll open it up to the audience for questions either directed at the panel or directed at myself. Sound good? You guys all set?
Yeah.
Okay. Ready. All right. I need to stop talking. Panel discussion. So let's see. So I guess just to get started, maybe you guys could describe just a little bit about your research, specifically kind of the limitations or gaps that you may see in the proteomics space, and if you want to wade into the waters of the Platinum technology, where you see Platinum fitting into that work.
Okay, happy to go first. So John Prensner, University of Michigan. I am a cancer biologist by training and a pediatrician, pediatric oncologist by clinical work. And so our interests are really on trying to bring cures to patients for children. And so how do I fit in here? I fit in here because I've developed an interest in the dark proteome. This goes by several names. Audience members might know in different ways, but these are essentially the small either microproteins or small open reading frames or retroviral derived proteins and their potential therapeutic importance in various cancer diseases. And so I've been active in that space in the research world and starting to wade the waters of proteomics, where I'm not really a trained proteomics expert, and I have a relatively rudimentary understanding of peptide spectra from a mass spectrometer. I can look at them and see a little bit, but not see too much. And so I got intrigued by QSI because it actually speaks the sequencing language that I think I understand quite well. And so conceptually, it shifts from sort of a PSM-centric universe to a sequencing read-based universe, which seemed quite adaptable for my, I guess, limited genomics brain, where my mass spectrometry is maybe weaker than many in this room. So that's how I kind of got into this space, and we are, in fact, onboarding a machine, as Joel mentioned.
All right, so I guess conversely, I would say that I'm a mass spectrometrist, and my laboratory studies a rare and fatal disorder called Niemann-Pick type C. Sometimes it's referred to as childhood Alzheimer's disease. And so we're really committed to trying to better understand how the proteome is changing in models of this disorder, as well as being able to identify candidate biomarkers that can be utilized for both therapeutic development as well as for clinical monitoring. So a lot of our work has more recently been focused on finding these biomarkers and then making targeted assays towards a larger scale analysis. One other aspect of what we're doing, so this disease arises from mutations in a large membrane protein that serves as a cholesterol transporter. And we're now starting to spend a little bit of time trying to better understand that protein, in terms of its structure and its function beyond just cholesterol transport. So those are some of the things that we're working on. And like I said, we're very much mass spectrometrists, but we're certainly open to bringing in other technologies and strategies towards our ultimate goal.
Hey everyone. And so my lab has been really interested in applying mass spectrometry-based proteomic approaches, bottom-up, middle-down, top-down, for understanding protein post-translational modifications, especially on proteins such as histones, but we're interested in a lot of other things as well. And over time, we've developed a lot of methods that are based on mass spectrometry to try to sequence proteins and identify and quantify these modifications. But yeah, we've definitely noticed that there's some proteins, some proteoforms, some approaches that still leave us some gaps. And it was really nice, Joel highlighted that paper that Neil had collaborated with Quantum SI, where they kind of show the combination of top-down and the platinum really are very complementary. Oftentimes in top-down proteomics, you might be able to sequence the ends of the proteins really, really well, but once you get towards the middle of the protein, it becomes harder. And the approaches on the mass spec side are getting better and better, but that's often where we see gaps in the sequencing. And so here, immediately right now, the platinum actually can fill a lot of those gaps. I see a lot of really interesting applications on the horizon as well as this kind of technology develops, not only in the proteoform or isoform kind of space. Stephanie mentioned membrane proteins as well, kind of notorious proteins to kind of sequence by any type of mass spectrometry approach. I see, potential extensions to other molecules that have been kind of challenging in the past, those that don't have a lot of charge, things like, longer MHC molecules and things like that. So I think those are some immediate kind of problems that this technology can, help with, as well as I'm very excited, about the future. On Sunday, if you guys were at that, future of proteomics session, we talked a lot about, how as a field do we move forward to get a lot of people involved, and how do we lower the barrier to enter into the proteomics field? Will there be a time where there will be a benchtop, instrument, whether it's mass spec or something else, that someone can just, with very minimal experience, not needing a PhD in mass spectrometry and a postdoc in mass spec, be able to, generate a lot of incredible, useful data. And I kind of see that here. I see, not right now, but I see the trajectory for this technology to one day hit that. So.
Thank you. Any other thoughts before we kind of move to the next? Okay. Yeah, so I think, so one of the other questions is sort of around the field of proteomics generally. I mean, I think there are a lot of tools for analyzing proteins. I think, what do you guys see as sort of the field of proteomics today as compared to what you envision for the field of proteomics in the future? Ben, you sort of alluded to it a little bit, but where do you think we go from here, and what do you think the role of technology is in that sort of future proteomics state?
Can I start? So, a few things that come to mind when you ask that question. The first is robustness. So I think it's really important. I mean, mass spectrometry, for example, has made great advances, right? But I think there are still challenges with robustness, and so I think for proteomics to be really embraced by more folks, that'll be important. And I also think a lot about speed, like throughput. And like I said earlier, introducing some of the work we're doing in my lab, it's just not feasible for us to sometimes think about running thousands of samples under some of the current timescales that we're doing. So those are the two things that pop in my head.
Yeah, I think those are great aspects to consider. I mean, again, kind of going back to things that were discussed at the Future of Proteomics kind of session, I do think a lot about how the genomics field has continued to evolve and everything that was done at the beginning to make sure that things were standardized and, data acquisition, data storage, things were kind of, developed to the point now that essentially anyone can get genomics data fairly easily. If they have their own sequencer or at a core facility, the data analysis is also pretty streamlined too. My lab, we always have students that want to complement the proteomics and add RNA-seq, and it's not hard to do that now. But conversely, if there's a biologist, if there's a genomicist that kind of wants to explore past the genomic side to do, proteomics or other types of omics, metabolomics, very, very difficult for a lot of reasons. And so I think if we're ever really going to be competitive with the genomics field for a lot of things, especially NIH funding, we need to get more people using proteomics. We need to be able to have benchtop instruments that biologists could use as easy as a qPCR instrument, as easy as to run ELISA or Westerns. If we don't have that, people are not going to use proteomics. We'll always have everyone here at US HUPO using mass spec and doing amazing things, but we won't have the kinds of numbers that we're going to need to be able to convince the NIH to create a study section that is focused on proteomics or an institute that is focused on proteomics. And so that, I think, is something, whether it gets solved by mass spec or some of these emerging technologies, is something we need to do. We need to be able to have an undergrad, first-year grad student come in the lab and within a couple weeks be generating high-quality, robust data. And so that is why I'm kind of very excited about this type of technology. And I also hope the proteomics field continues to move in that direction. I do think there's some opportunities there as well. I think there's always going to be a need for proteomicists to push for the cutting edge, to do the really complicated experiments, those things that are incredibly innovative and do require a PhD or a postdoc in proteomics or mass spec to do. But then at the same time, I think there's a lot of opportunity and a lot of need, and especially if we're going to continue to grow our field and get the public to understand what proteomics is, we need to get as many people at that basal level to be able to generate very good quality data on proteomes or single proteins or at a level that allows them to answer their questions. Maybe not this like expert level that gets to the millions of proteoforms, but to a level that will, get them the data they need to, answer their questions and continue their studies. And so that's kind of what I'm thinking could be a great opportunity for a lot of these emerging technologies to complement the proteomics field and help it grow as well.
So I think, Ben, you kind of took the words out of my mouth. I could,
Mine
I guess,
too.
sit before you here as, I guess, living evidence of somebody from the genomics field, and that's me, starting to venture into the proteomics field through an emerging technology. And the concept that even if I had the money, my lab would buy a mass spectrometer, that's kind of a non-starter conversation. We wouldn't know how to use it. The data analysis is too hard. We're just not trained that way. Conversely, I think it's been nicely said, as a clinician, many programs are now training their clinical oncologists to run their own RNA-seq BAM files or whole exome sequencing as a core skill that as an oncologist you should be able to know how do you get to that mutation in that cancer in your patient sample. Now, of course, there's complexities that go beyond a first-pass analysis, but that type of thinking is now being democratized across clinical medicine, and it's not being democratized for mass spectrometry or proteomics. And so how do we bring these technologies to people? You could sit here and make the argument, why do you need to do whole exome sequencing to find point mutations in KRAS? It's an expressed oncogene. Couldn't you do proteomics, find that same G12C mutation and also the PTMs of the signaling pathway, all from one experiment? You could make that argument. And so how do we get there to try to bring that additional data-rich information to people like clinicians who are not going to be experts in mass spectrometry? And so that's, I guess, echoing Ben, I'm quite excited by thinking about emerging technologies that can help us get there in medicine.
Couldn't have been a better segue into my next question. So, I mean, we're talking, I recognize we're at a proteomics conference filled with people who are experts in proteomics, but when we start to talk about multi-omics and sort of, you guys both sort of organically mentioned it, right? What are do you have specific challenges or opportunities that you see in making multi-omics data more accessible and actionable? John, you kind of referred to the barrier to entry for some technologies. Certainly it's always going to be needed, not going to make that argument, but what are the challenges and opportunities? Like, I always think of to kind of rip off Simon Sinek, like, why, right? Start with why, right? And I thought Neil Kelleher did a great job earlier in his panel session where he sort of told us the why. He said there's a lot of proteoforms that likely contribute to disease, but you can't read out at the genetic level, right? Maybe, so if you guys could just elaborate on some of the challenges behind moving toward this proteomics-led multi-omics space, challenges and then maybe specific opportunities. Like, do you guys come across in your research specific opportunities where you're like, man, if I leaned harder into a certain technology, I'd be able to unlock some findings that I didn't have before, or places where you see, man, if this paper only used proteomics, right, we'd be able to get more information out to you guys.
Maybe I'll start with this. I'll start with this question of ways to advance the next step in the proteomics field generally. Although I'm not an embedded member of the proteomics community, I liaise with a number of folks. So in my role as the Stark Proteome person, we work quite extensively with members of HUPO, including members who are organizing the next World Congress. We work with Peptide Atlas and the folks there, and have a number of sort of joint projects in that space. And so I'd say that the genomics community, I think, has done quite well to disseminate this, is to have pretty clean, nice packages that people agree upon work all the time to analyze your data. And I think that in the proteomics space, one way that can help advance this multi-omics integration is having really standardized community accepted ways to do this kind of all the time across platforms and not have to have platform specific considerations and things like this. None of which I'm particularly expert on to comment on. I just sort of observe it, I guess, as an outsider. And so, I think maybe one of the asks to kind of move this forward is thinking about how do you make this really easy for people? And even if the data acquisition is hard, you can't afford your own machine on your own benchtop. Once you generate that data file, how do you make it really easy for people across labs to do that same analysis and get the same result with relatively little kind of onboard training? So I don't know if that answers part of your question about these multi-omics integrations, but I think that's one thing the genomics community has done quite well at this point.
So I think when you said multi-omics, I thought of things a little differently.
Okay. That's good. Different's good.
I think that, I think it's been said, genomics is a really strong field. And it's kind of natural to think about, the genome and the proteome, but I kind of think about the other omes. And to me, I think the biggest opportunity is data integration. And I don't think that we're particularly great at it, actually, which is, if proteomics is your core starting point, whatever that other ome is, I think there's a lot of opportunity for growth in integrating data.
Yeah, not a lot to add. I think John and Stephanie really kind of hit the things. The barrier to be able to do a lot of these different measurements is tough. Data integration approaches are not quite there. They're kind of getting there, and some people are working well on this. But the need is immediate, right? Because there's been some really great talks already at this conference where people are showing that if they just look at protein expression or they just look at phosphorylation events, it's not the whole picture, right? They have to incorporate metabolomics. They have to incorporate other types of omics, the types of analyses. And great talk this morning about how even being able to acquire the data for proteome, metabolome, lipidome in the same experiment. But then the challenge of the data integration is real and very, very difficult. But I think that's where the field is moving. I mean, I think, proteomics is moving towards, doing all these things that genomics can't do. Just looking at genomic mutations alone, just looking at RNA-seq, mRNA expression alone, it tells you something, but it doesn't tell you the whole picture. And many times it may not give you the right picture because of a lot of processing events that happen at the RNA level and because of modifications and protein complexes that affect enzymatic activities and other types of functions. And so, I would say, just to kind of echo, I think that the barrier to get all of these types of experiments is quite tough. Maybe it might be, especially for someone outside of the field, I think that's really difficult. And then the data integration is also one of those things that is difficult as well. So one of the easier experiments might be to do a protein expression and the mRNA, right? For someone, it seems like John was saying, even trainees are starting to get trained in how to do RNA-seq, right off the bat. If there was the protein equivalent of that type of experiment, then our trainees would be able to get RNA-seq and protein expression kind of training, and that would be the first steps. And then they could kind of keep building and building, adding more multi-omics, getting more interested in potential mass spectrometry and other experiments to hone in on metabolites and other things. So it's a great opportunity for sure.
So we've defined sort of the ideal future state of a proteomics space where more people are doing it, it's more accessible. What are the things you think that the field needs to do, and this is not necessarily related to Platinum or what Quantum SI does, but just to make sure that the tools are available to a broader range of researchers, whether it's limited resources or geographically or whatever. Like, what do you think the things that the field should be thinking about now to ensure that ideal future state has access to these sorts of technologies? There's probably several things, I think. But I think, first off, just being able to acquire the data well, knowing that you have high quality data, statistics to be able to understand your data and assess it. I think all those are probably lacking a little bit in terms of training for younger scientists, especially through the programs they're going to. Sometimes when they go to a specific lab, they will get trained on how to do this and that and whatever. But I think education-wise, we probably need to figure out how the proteomics field
can help other life sciences understand that, besides knowing the foundations of molecular biology, biochemistry, things like that, they also should be, learning like, foundations in statistics, computational analyses, coding. I mean, these are some things I know some programs are doing, but I don't think it's universal. And I think with all this large scale, we're talking omics, you're going to be generating a lot of data. There's got to be, a way to make sure that all of our younger scientists are, handling the data properly, analyzing the data properly, getting the right results, from this type of data. So I think there's opportunity that, maybe as a field we can kind of, all of us are in different institutions or in other societies, we can kind of try to emphasize how important this is, because a lot of people are using omics, starting off with genomics, but we want them to use more proteomics as well. So I think that's one thing. I think the other thing that I think about too is just, and we talked about this a little bit at the Future of Proteomics workshop, is just what, educating the public about proteomics, right? Everybody kind of knows what a genome is. If you grab some random person off the street, they know what DNA is, but, do they know what phosphorylation is, or, proteoform or proteomics? Probably not. Or as someone was saying, if they think of proteins, they're probably thinking of like nutritional supplements or something like that. And so I think there's also needs to be a great effort to advocate for our field to the public, as well as, of course, to Congress and anybody else who will listen that are involved in funding decisions and things like this. But also just to educate that, this is the next kind of, building off of the Human Genome Project. Now we have this really great opportunity to understand how the whole cell works, and a lot of that is going to come through proteomics or metabolomics.
Sure, yeah. Let's start the Q&A.
Struck me listening, and I've read about the company previously, that you take for granted everybody understands what quantum science is.
Okay,
And
yeah.
you take for granted everybody understands what platinum is.
Okay.
I know it's a natural element, but I don't think that's the meaning here exactly.
No, yeah.
So maybe it would help if we all had a common understanding of those two keywords in
The quantum,
presentation.
the quantum piece? Is that, yeah, so really, oh yeah, so sort of a history test on the company. I think quantum is really derived from the fact that we have a really proprietary chip technology. And so I think it relates more to the engineering of the technology. And platinum is just the name of the system. And so it doesn't have anything to do with necessarily quantum technology, so to speak. It's more just about, the packing of the technology into the chip and the engineering of the technology.
The word that conveys excellence,
Sure.
I guess.
High-tech engineering, for sure. Lots of patents went into the development of technology, yeah. Thank you.
Do you have thoughts? Go for
I
it.
have one.
Yeah, go for it.
One comment that I would have, just to follow up with what Ben said on this question on how to make proteomics more accessible globally and to all populations. I would continue to advocate that the proteomics community continues to work on data dissemination through public repositories, whether or not that's Proteome Exchange or PRIDE. I think it's great that these exist. I think they can always continue to be revised, improved, and in ways that facilitate reproducible research, in ways that facilitate other researchers downloading that data and being able to use it, because there's such a wealth of public information and public data. Reaching the global research population or global research public really is facilitated through these. So I think that could be a priority. And when you think about emerging technologies, whether or not that's next generation protein sequencing or other emerging technologies, how do those emerging technologies get their data into those places too? So that other people across the world can start to say, you know what, I might not be able to buy my own QSI machine right now, but we can go and download this published data set from this other lab that put it up, and we can start to learn about these data and help make innovations and discoveries across the field as well.
Any additional? No? Good? Okay. So we have 5 minutes left, so I think, to wrap up, hopefully you guys found this session useful. I appreciate you guys spending the hour with Quantum SI, but also with our lovely panel and all of your colleagues. So if you're interested in learning more about Quantum SI, again, we're at booth 11, happy to chat with you guys about any specific questions you might have about the technology or the applications. We also have 3 more posters. We presented 1 poster yesterday with 3 more today. Mine's poster 24, and forgive me, my colleagues, I forget what the posters are for them, but they're all in the same area. Yeah, would love to have you guys come by and ask some questions. And so we have probably, at this point, 5 minutes left. We could take a few questions if you'd like, directed at myself or the panel, or we can break early. Yes? Yep, you. Yep, go ahead. Yeah, yeah.
Harris is from the Complex Carbohydrate Research Center.
Okay.
Can you comment on blood phosphatidation?
Sure.
Looking at the sites of blood phosphatidation or
Yeah.
other post-translational modifications.
That's a great,
Within
yeah.
your system.
Yeah, so what I can say is the kinetic signature that I talked about, that sort of on and off binding, is sensitive to sterics. So there's a sequence dependence to how that binding happens. So what is the amino acid upstream and what is the amino acid downstream? LAF will look different than LIF, will look different than LDF. And by extension, post-translational modifications generally have an impact on the kinetics in the surrounding area. To the specific PTM, so this is an active area of investment for us. We've looked at phosphorylation, we've looked at arginine methylation, we've looked at citrullination. In those cases, we tend to see an impact on the kinetic signature. You do have to do some training of the system to make sure you understand what is going on. And that, so maybe run synthetic peptides, that sort of thing, right, to really help level set yourself. But I would say the glycosylation specifically is a pretty complex one, I would say, because of the diversity of the species that you can have. I don't have any data to share on that, truthfully, but I think there probably is a place for the technology in that space. How deep it can get with recognizers that recognize amino acids versus specific glycosylation species, I can't say. But yeah, does that answer your question? Yeah. Yeah, thank you. I appreciate that. Yes?
Yeah, you've been talking about the democratization of proteomics
Sure.
versus genomics, and there's a really nice diversity in the panel in terms of mass spec knowledge. And I'm just curious, you mentioned that for glycosylation, there's a little bit of a ramping up to understand that,
Yeah.
analyze that. For the panel who have this knowledge of both mass spec and how long does it take for someone who's naive to the technology of quantum SI to really get to the point where they can understand the data in the way that you can understand mass spec?
I think it probably would, yeah.
I can start with that. So we have done our first initial experiments in collaboration with the company and are onboarding the machine, although we haven't actually pressed the go button in the lab on the machine yet. So what I can say is that the sample prep appears quite simple, and so it's not anything challenging. With the data generation, at this point, I think the company's bioinformatics team has been quite usefully involved in trying to help us make sure we can understand and process the data through the cloud. And so it's actually quite collaborative of an environment. The types of data reports that we have received back as files have been, to me, quite intuitively understandable. And so I can just share one very simple first experiment as kind of like a, if this doesn't work, then I'm a little worried. We express GFP in HEC293 T cells and say, do we find GFP, right? And indeed, you do find a lot of GFP, and it looks good. And so that was one of those, sort of go, no-go experiments. If it fails that test, then it's like, oh, why didn't you find the GFP? And that data report was quite easy for us to navigate. I don't know if that fully answers your question, but that's been our experience thus far.
All right, maybe time for one more question. Now, I'm happy to hang around after and ask questions. Otherwise, I'll give you 2 minutes back on your day. Yeah.
Yeah, you mentioned that you were in the middle there. We're working on membrane proteins, so I was curious about the buffer requirements for input of your protein. In this handle detergents or anything that, mass spectrometers would,
That is
paint over? Is this a little more,
very good question. Work is ongoing to define the common buffers that people use and their compatibility with the technology. I will say that anecdotally, I have run membrane proteins on site at clients, and it has generated sequencing results. Broadly speaking, there are specific components that we can get into offline. I'll actually have to check a reference, truthfully, to make sure. But we do not think that detergents at reasonable levels will be as disruptive as they might be for other technologies. But of course, our recognizers are proteins, right? And so, of course, at some point you have to have functional proteins. So I think it warrants a deeper conversation. But yeah, it's sort of a high-level answer, and I recognize that, but happy to take it offline and chat deeper. Yeah. All right, well, with that, I want to thank you guys again, and we'll hang out for a little while and answer questions if you have them, but please enjoy the rest of the conference. Thank you. Thank you guys so much. Thank you so much.